Good morning, boys and the girl. Today we continue with the chapter Biomolecules, and for your memory's sake, I would like to reiterate that this chapter will be the last chapter for Term 1 exams. As all of you know, CBSE is conducting your board exam 2 times. So that is Term 1 in the month of November, December, and Term 2 somewhere in March. So as part of the November-December Term 1 exam, already we have completed around 5 chapters, and this will be the last chapter, Biomolecules chapter. Okay, and I will forward the syllabus of the Term 1 and some MCQs already I have forwarded to you. I hope you are all practicing it. Keep on practicing MCQ, multiple choice questions. See, it is easy to write descriptive questions. You can write pages and pages, but when it comes to MCQ, you have to have logic. Okay, so develop that logic and critical thinking, then only you can answer. Plus, if you score 50 out of 50 here, then it will be very easy. In that, 35 marks theory will be there, 15 marks practicals. Maybe next month or so you will be coming to school back physically, then we will start our practicals also along with your 12th class, right? So going ahead, we were talking about lipids. Lipids are, it will be offline, no online. Yesterday you must have read in newspapers, CBSE has clearly said that it will be offline. Exam will be physical exam. You have to come. Yeah, you have to come to school and write. That is the thing. No online. Right? So we are planning to call you back, and before that we have to gear up for all the facilities, everything, no? Then the school will take a decision and call you. Meanwhile, what you have to do, first get familiarized with the syllabus. What are all the chapters that are there for Term 1 exam? What are the practicals that you have to do in biology? So as other subjects, physics, chemistry also, right? Find out. Go to CBSE website, log in, and you know the syllabus, 11th biology. I am posting it in the group, you can go through that. And then most important one is you getting familiarized with MCQ pattern. There are varieties of MCQs, like straight, one question, one statement, and you have alternatives A, B, C, D, E. So you rule out the wrong ones and arrive at the right one. That is one pattern. Second pattern is case study. One story will be given about a concept, right? And based on the story, there will be questions. And don't think that answers will be there in the chapter or in the paragraph that is given. So you have to analyze that story and then answer the questions. So you really need to train yourself for this. And the third one is, there is a pattern called assertion and reasoning. 2 statements will be there. Statement 1 is assertion. Statement 2 is reasoning. And the multiple choices will be like assertion is right, reason is wrong, reason is right, assertion is wrong. Assertion is right and reason is the correct explanation of assertion, and sometimes reverse. Assertion is correct but reason is not the correct one, not correct explanation for the assertion, like this. So you need to train yourself for this. Keep on practicing. I will be posting lot of questions like this, MCQs. Oh, don't waste time, right? Coming back, yeah. No, maybe for your batch, your Corona batch, no? It's because of Corona, last year and your batch. Now, once things become normal, I think the CBSE will switch over back to the original course, right? Or maybe they continue, because this will help you in your NEET exam, UPSC, ND exam, all this, no? Because most of the competitive exams are in MCQ form. So it will be a good practice for you, right? Most important one is, be it any type of question, you must understand the concept. Once concept is clear, let the question be in any form. How does it matter to us? Isn't it, Naveen? Yeah. So understand the concept, right? And then we are going ahead with the lipids. Last time we talked about the simple lipids, and lipids are nothing but fats, we said, and the simple lipids or simple fats will have all what we call single bond, carbon carbon, isn't it? And we said that any lipid will have two chemical molecules in that. One is the fatty acid, another is the glycerol, isn't it? Any fats contain two substances, glycerol plus fatty acid. And there will be a chain of fatty acids depending upon whether it is a simple lipid or compound lipid or derived lipid. So, number of fatty acids will be different. And now we go to the second one, that is compound lipids. So, structure of the cell membrane, you know it already. Fluid mosaic model, some of you have drawn beautiful diagrams also. In that, if you recollect from your memory, we said that there will be bilipid layer. Bi means two layers, see, lipid bilayer, as you can see. And in between that lipid bilayer, you can see varieties of proteins. Some are in between, so some are outside. We say intrinsic proteins, the one that are accommodated in between. Some are external, some are transport proteins, okay, as you are seeing, and also presence of carbohydrate chains, you can see. And that's how we said that our cell membrane is made up of carbohydrates, proteins, and fats. And fats are in two layers. So we said lipid bilayer. Recollect from that previous chapter, isn't it? And then compound lipids are the esters of fatty acids. You know, you have studied in chemistry esterification. So these are esters of fatty acids and alcohol with additional groups. Now, when we say this phospholipids contain fatty acids plus glycerol plus, as the name suggests, phosphate. So they are found in cell membrane, in our skin. Example he has given, lecithin. You can see the presence of so many structures, double bonds and phosphate groups. Okay, so the structure is little bit higher version. Hence we are using the word compound lipids. And then derived lipids, third one. These are the products of hydrolysis of simple lipids. Hydrolysis, hydros means water, lysis means breaking. So hydrolysis is process of breaking with the help of water molecules. Hydrolysis of simple lipids and compound lipids. The structure is our very famous cholesterol. And mind you, we have a tendency that whenever we hear this word cholesterol, we are tempted to take it as a negative one. I said that cholesterol can be both positive as well as negative. In terms, we say good cholesterol and bad cholesterol. Good cholesterol is very essential for us, and bad cholesterol is to be eliminated because it blocks the arteries. So there will be problem in flow of blood. Hence the bad cholesterol is to be removed as and when time permits. Let me check whether it is yes. Right. Now they can join directly. Moving ahead, we have next the carbohydrates, and the best examples are sugars. As the name suggests, carbohydrates. Carbon, hydrogen will be present along with the oxygen. Yeah. Yes, somebody, please mute. Please mute. Who is this? Yes. Sachin. Yes.
Yes, sir.
Sachin, there is some voice.
Sorry.
Okay. Mute yourself. Right. So, sugars, when we talk of like the common sugar that we eat also. And we said the other day in DNA, RNA, also sugars will be there. We said the sugar that is present is pentose sugar, ribulose. RNA is ribulose nucleic acid. And if you remove one oxygen atom from that, it is deoxyribulose. So deoxyribulose sugar is present in DNA, ribulose sugar is present in RNA. So that shows the importance of sugars. Even in our genetic material, sugars are present. And I'm sure today morning when you got up, all of you had tea, coffee, and your mummy must have added sugar. So without sugar, the tea and coffee may not be liked by you. So sugars play a very vital role. And what about sweets, Pallavi, that you are all eating? Invariably, sugars. And what about the rice that we South Indians are all eating? Again, carbohydrate. What about the bread, bun, biscuits? All these are carbohydrates. So basically, sugars are sweet and water soluble carbohydrates. And what is that they are made up of? As I said, CHO, carbon, hydrogen, oxygen, in the ratio of 1:2:1. And example is glucose and ribulose. Just now I said, you can practice this. Glucose C6H12O6. And we said C6 carbon atoms. Look at that, CH2OH we are writing on top. And in all the corners you have to put one carbon. Carbon 1, 2, 3, 4, 5, 6. And one side you can see on the right side up O is written, oxygen atom. So don't put there carbon, it's oxygen. So C6H12O6. And similarly ribulose, C5 it is. Okay, ribulose is C5H10O5. See the structure, O has come centrally because it is a pentose sugar. Penta means 5. So just draw these diagrams for your convenience sake. Then in DNA, the most important element, be it DNA or RNA, is nitrogenous bases or nitrogen bases. And basically, you must understand that nitrogen bases will be of two types based on their structure. See, you can see some of them are single ringed structure. One ring only will be there. Okay? And some are double ringed. Two rings are there. Just watch out. Double ringed structures and single ringed structures. And they are so called because of the presence of nitrogen. That's why nitrogenous bases. And based on their structure, as you said, double ring and single ring, you can see here presence of nitrogen. And there are two types of nitrogenous bases. One is called purines, another is called pyrimidines. See the spelling, purines and pyrimidines. And in turn, purines are two types, adenine and guanine. How to remember, Naveen, in the exam? Purines spelling starts with P. Purines, purines. Adenine starts with A. Guanine starts with G. So PAG. Reverse you take it. GAP, gap. So remember that abbreviation GAP. Guanine, adenine are purines. So remember that abbreviation gap. And pyrimidine, PY, it also starts with a P, but you pick up PY. And 3 are there under pyrimidines. What are those? Cytosine, thymine, and uracil. Right? And particularly last two, thymine and uracil. Thymine will be present in DNA, whereas when it comes to RNA, it is uracil. That is the difference, one of the differences between DNA and RNA. Okay? In place of thymine, uracil will be there. And how to remember? There we said gap, guanine, adenine is equal to purines. Here, instead of P, you pick up PY, pyrimidines, and cytosine C. So pick, PYC, pick, and UT, Union Territory. In your geography you are studying, no? Goa, Delhi, you are saying, no? Union Territories, Daman, Diu, Pondicherry, like that. So Union Territory, you remember UT. So pick UT, or you can form like this very conveniently for your memory's sake, your own abbreviations, right? And the structure will be like this. Look at that. Uracil will be like this, which is a pyrimidine, and adenine is like this. So draw the diagrams, at least twice you draw the diagram so that you remember it, right? So purine and pyrimidines. Purines are double ringed structure. Pyrimidine is single ringed structure. Okay. So keep practicing. Keep this both the structures to be practiced, and then we come to next molecule, that's called nucleoside. Okay, nucleoside. This is also part of nitrogenous bases. What is a nucleoside? This is a very common question. So nitrogenous base plus sugar. Okay, and how to remember nucleoside? What is the spelling? NuCleosides. Correct now? In that, N, yes. N stands for nitrogen base, S for sugar. So in that word only you get the answer. If someone asks you what is a nucleoside, in the question only answer is there. What is that? Nucleoside. N, S. N for nitrogenous base, S for sugar. N plus S is equal to nucleoside. So look at that. How many types of nucleosides can be present? Nitrogenous base, if it is adenine plus sugar, adenosine. If the nitrogen base is guanine, guanine plus sugar, guanosine. If the nitrogen base is cytosine and when it combines with sugar, it forms cytidine. And if it is thymine, combining with the sugar, thymidine. And in case of RNA, uracil, because uracil is present only in, I said, RNA. Uracil plus sugar, uridine. So these are all the nucleosides: adenosine, guanosine, cytidine, thymidine, uridine. All these are nucleosides. And how to represent? Look at that. This is the way. Adenosine and uridine. Sugar plus nitrogen base. Combination you have to draw. And then we go to the next one, continuing with the nitrogen base only. Top we said nucleoside. Now we are talking about nucleotide. Nucleotide. That is, we said NS, nucleoside, nitrogen plus sugar. So what is left out in DNA molecule or RNA molecule? It is phosphate. So nucleoside plus phosphate is equal to nucleotide. I repeat, nucleoside plus phosphate is equal to nucleotide. I hope you are getting it, Naveen. I hope you are following me. Pallavi, Pallavi is here. Gowda. Following? Okay. Now, nucleoside plus phosphate. Look at that. Nucleotide. That means why Naveen will be thinking why Sir is telling all this. If someone asks Naveen, why you are appearing like this? What is the answer of Naveen? Why Naveen is appearing, how he is appearing, why he is appearing. What is the answer of Naveen? Yeah, see, genes. Naveen says, sir, my genes make me. Now, next question by Pallavi is, where are those genes? What are genes? Then Naveen will tell, gene is portion of DNA. Correct? When we say portion of DNA, we should know what is DNA next. So DNA is made up of nucleotides. So Naveen says DNA is polynucleotidal chain. Polynucleotidal chain. So then Pallavi will ask, what is a nucleotide? There comes Naveen. Nucleotide is a combination of three chemical substances: nitrogenous base, sugar, phosphate. Correct? Nitrogen base, sugar, phosphate. So NSP, N for nitrogen base, S for sugar, P for phosphate. NSP is equal to nucleotide. That is one nucleotide. We said polynucleotide. Poly means many. Many nucleotides are present. Hence, say DNA is polynucleotidal chain. I hope I made it clear. When we say polynucleotidal, many, what are those many? So one is adenine, nitrogen base, plus ribulose sugar, plus phosphate, that becomes adenalic acid, which is a nucleotide. Second, guanine plus sugar plus phosphate, guanilic acid. Second one. Third, cytosine plus sugar plus phosphate, cytidilic acid. That is the third type of nucleotide. And the fourth, thymine plus sugar plus phosphate, thymidilic acid. And finally in RNA, uracil. I said uracil nitrogen base, and it is present only in RNA. So uracil plus sugar plus phosphate is equal to uridylic acid. Uridylic acid. So when we say DNA is a polynucleotidal chain, it is made up of many nucleotides such as adenilic acid, guanilic acid, cytidilic acid, thymidilic acid. Of course, if you ask what is RNA, then Naveen has to say uridylic acid. And in which place this uridylic acid will come? In thymidilic acid. Thymidilic will be present in DNA. In place of that, uridylic will come if it is RNA. I hope it's clear. Now, we come to the DNA molecule. Once we know that what DNA is made up of, again I repeat, if someone asks our Pallavi, what is DNA? Normally, many times they will ask you, no Pallavi, what's your DNA? Okay? For Sunday school boys and the girls, the people in the society will ask, what is your DNA? What is so unique about you being a Sunday school student? Isn't it? So here, your DNA is polynucleotidal chain. And if you see carefully, This DNA molecule is in the form of twisted ladder. You know, Naveen, ladder, Nichenege. All of you climb along with the ladder and reach the top of your house, Hosla house, Vijayanagar house, like that. Suppose one side Naveen holds the ladder, other side Raghavendra holds the ladder and twist it. Then you get this shape. So this shape is discovered by two professors. We are thankful to them. Whatever technology we have developed, whatever concept that we understood about human beings, about other organisms, plants and animals, in fact all living organisms, we must be always grateful and express our gratitude to two professors, Professor Crick, Professor Watson. It is they who discovered this, and we must be proud that in this there's one Indian scientist also, Hargobind Khurana, right? So basically DNA is a double helix structure. They use the word double helix. Means when ladder Nichenige is twisted, it forms this. And when we measure, like normally we measure the distance from Bijapur to your hometown in terms of kilometers, right? Here the distance is measured in terms of angstrom units. Again, angstrom is a scientist. It is named after his name, angstrom units. So the 1 angstrom, specific angstrom like 34 degrees angstroms will form a gene, and it means physically we can say almost 1.5 loop, right? 34 angstrom units. And at present, yes, Raghavendra.
Sir, why DNA can't be straight like ladder? So what is significance of its being twisted?
It is because of the bonding that takes place among the 3 chemical compounds present in a nucleotide: sugar, phosphate, nitrogen base. You see, it is the combination of 3, no? And whenever there is a combination, you know, energy gained or energy lost. Your chemistry teacher must have told you, exothermic, endothermic, isn't it? So here, because of that bonding, the twist will be there. Look at that. This is now the bonding. There are 2, if you observe, one blue ribbon-like structure and one orange ribbon-like structure. Correct? These blue and orange ribbons are made up of SP. SP means, Raghavendra, Superintendent of Police. Isn't it? Yeah, SP is sugar plus phosphate. So the ribbons are made up of SP, sugars plus phosphate. And you can see, like in ladder, steps, horizontal steps you are seeing. Okay? Those horizontal steps are nitrogenous bases. I hope you are able to understand. 2 vertical lines in the ladder are made up of SP, sugar plus phosphate, sugar plus phosphate. Connecting the 2 vertical lines, horizontal lines are there. And those horizontal lines are nitrogenous bases. And already we discussed, nitrogenous bases are of 2 types, purines and pyrimidines. And purines are double ringed structure, pyrimidine is single ringed structure. And there is a base pair rule. What is the base pair rule? One purine combines with another pyrimidine. Not purine, not pyrimidine. It is purine versus pyrimidine. Bonding is always purine versus pyrimidine. So, if let's say purine is adenine, okay, and it always combines with thymine, AT, or it can be, Raghavendra, TA, thymine combining with arginine. And second one, guanine combines with cytosine, or vice versa. Cytosine combines with guanine. So the horizontal steps that you are seeing, let me expand these. These horizontal steps that you are seeing, they are made up of nitrogenous bases. So vertical ones are SP, sugar phosphate, Horizontal ones are nitrogen bases, purine, pyrimidine, pyrimidine, purine. So this is the inner thing of the DNA. And when we defined DNA as polynucleotidal chain, what is a nucleotide? We said SPN, sugar, phosphate, nitrogen. So you have many SPN both on left side and right side. Correct, no, Raghavendra? Are you able to follow? So when you have the SPN, one SPN is equal to, Raghavendra said just now, nucleotide. And look at that, how many nucleotides are there within these two loops? Hence, Raghavendra defines DNA as the polynucleotidal chain. So you need to practice this. Soon after the class, when you are free, these structures you have to draw. Look at that, ringed structures. These are all, your chemistry teacher must have told you. Benzene, right? Same is the case. So please draw this, and in our WhatsApp group, put a thumbs up. I understand that you have practiced it. If I am not getting a thumbs up from Pallavi, it means she has not done it. Right? Today I am expecting all the children putting thumbs up in the WhatsApp group. Right? Keep practicing, and I will proceed with the next topic in the next class. Take care. Bye. Yeah, please. Gene therapy?
Genes are made up of proteins or some other.
Gene, just now we said, what is a gene? Portion of DNA. And portion of DNA is made up of SPN, sugar, phosphate, nitrogen base. So basically gene is nothing but SPN combination. Correct? No? And these you are right, these transform, undergo some change and form protein. That we will talk in 12th class. When you come to 12th class, we will talk about protein synthesis. But for that protein synthesis, this is required. This is the basic requirement to synthesize a protein. And your guess is correct. Ultimately, it is the proteins that will express characters in us. And what we are is ability of our characters. Right? Okay. Bye then. Take care.